dementia researcher with a blog and a rating.
My first clinical trial, lessons learned and surprises along the way. I was involved in my first clinical trial in 2005 when a pharmaceutical company asked my colleague, a consultant old age psychiatrist who had experience in trials, if he would be willing to pull a team together to run a clinical trial. This was long before there were disease-specific clinical research networks in the area. Crucially, this trial required the research staff to have expert knowledge about dementia. We were gathered as a group to discuss whether we could do this. All of us were primarily clinicians, 2 medics and 2 nurses. I was the designated clinical trial coordinator, a role I had never done before. We were able to screen potential participants from both of the medics' clinic lists and apply the inclusion and exclusion criteria to the potential participants. We told the pharmaceutical company, or the sponsor, that we could recruit 4, and that is the number we ended up recruiting. All of us attended our first investigator meeting in Vienna, which was a complete culture shock to us NHS employees, as this was a no-expense-spared meeting which had us each being handed a €50 note in an envelope on arrival, as our plane was delayed and we had missed dinner. The next evening, seeing the Vienna Boys Choir being taken to lavish restaurants with copious amounts of food and drink, remember this was 2005, in return for our compulsory attendance at the most tedious lectures about how to perform the most basic of trial activities. Due to compulsory consumption of all that free alcohol and food, the 8 a.m. to 5 p.m. days in enclosed, no fresh air or sunlight allowed rooms were particularly hard going. Lectures included how to perform the mini mental state examination, which as clinicians we knew very well. The difference was how the sponsor wanted us to perform it, consistency being the key, for 2 hours. Being marked on our ability to perform the clinical dementia rating scale and what it means to be a blinded rater for 5 hours. Learning all about the ADAS-COG, the Alzheimer's Disease Assessment Scale, cognitive subscale. I do not recall how many hours, but it was many. The most tedious lecture I can still remember vividly, 20 years later, as the one where they instructed us on how to put blood bottles in a bag and then to use the correct labelling for the bag and to put that bag into a box and send it off with a courier. There was a separate lecture for handling dry ice. I didn't realise until the study was underway that this would become the bane of my research life. We returned to base and worked out who would be doing which scales and who was the blinded rater. The company approved our choices based on the results of the tests back in Vienna. We then organised our first screening visit. This was where things started to go badly wrong. We vastly underestimated the amount of time needed for each screening visit, and if we could do it again, we would have only asked one of the participants to come along, rather than all 4, staggering them by 1 hour. It was a complete disaster and ran way over time. We hadn't ordered food for anyone. There was no time to offer anything other than a cup of water. The courier arrived to pick up bloods that hadn't been taken. The participants were waiting for hours to be seen. We, the staff, were completely overwhelmed with the amount to be done for each participant and their accompanying carer. Our subsequent visits were much better organised, and we were able to carry out the visits and assessments in comfort for everyone concerned, participants and staff, as well as organise all the assessments and write up following each visit. I mentioned earlier that dry ice was my biggest headache. All the samples which had to be sent on dry ice were going to a lab in Switzerland. This required knowledge of airway bills, storage and handling of dry ice, but most importantly, a hotline to the carrier. Ice had to arrive on the day of visit to the correct hospital and to be picked up a few hours later. As we were using 2 community hospitals in more rural locations to see participants, the ice had to be delivered and picked up there. Sometimes there was no receptionist and the package would be returned, so no dry ice. Sometimes the carrier would deny all knowledge of the hospital's existence, and there would be no dry ice. Sometimes it would not arrive on time, or the courier would arrive too early to pick up the package and would refuse to wait. The most memorable occasion, the ice had arrived the day before and had shared the night in a hospital morgue with an unfortunate companion. What we learned: overcompensate for time, allow breaks for the participants and staff, ensure there are refreshment facilities for everyone. Organisation is key. The right staff doing the right assessments with the right participants and the right paperwork in the right place. Check and check again with the carriers that they know where and when the dry ice is to be dropped off and collected. Do not expect lavish receptions at the investigator meetings. This has never happened since.
Thank you for listening. Join the dementia research bloggers and share your own views.